You're watching CTOM TV. Knowledge is power.
Yeah, overexpression is a very important thing to understand, and it is a huge factor in lymphomas. So, and many different, cancers, unique cancers. So, basically, our body has the same genome in every cell. However, if you notice, our body has many different types of tissues and many different types of cells. I mean, our skin looks very different from our hair or our eyes, our bones, and every person's skin and bones and eyes and features look different. And so the way that happens with using the same DNA in our bodies, what happens is there's differential gene activation in every cell. So the way our body uses the same genetic template to produce a bunch of different tissues is by turning some genes off and some genes on in different tissues. So there is differential gene activation and expression in different tissues. So this is an important thing to understand. So what happens in cancers is certain genes that are not supposed to be turned on get turned on and turned on to a much higher degree. And other genes that are not supposed to be turned off get turned off, such as tumor suppressors. So overexpression in different tissues is typically what we call an epigenetic modification. So epigenetics is what controls what genes are turned on in different tissues. So theoretically, we do have we have the genes to grow a tail if we wanted to. They're just turned off in our bodies. So, we have a lot of different genes that we don't use. Well, we probably use all of them at some point in time, but have a lot of extra DNA and extra genes. So basically what overexpression is when a gene is turned on to a much higher degree. So for many years, we didn't have the ability to really do anything with that because, we have 25,000 different genes and some of those genes have multiple different transcripts. So, that's just a massive number. But now with next generation sequencing, we can actually look at 20,000 different genes. So what we do, and this test has only recently been available for clinical analysis. It's been used in research for a long time, but, we haven't really been able to use it for treating patients until recently. And it's been a game changer for us. So what we have is this test that will isolate the RNA, and this is what it looks at. It looks at the RNA, actually counts the amount of RNA and compares it to a normal tissue. So we have this test that looks at 20,000 genes, looks at the expression of these genes and compares them to what is normally supposed to be expressed in your normal tissues. So if you have, for example, ovarian cancer, then we would compare the expression of the ovarian cancer genes, 20,000 of them, to the expression in normal ovarian tissue. And that tells us this gene is overexpressed, therefore this drug is going to be important for you.
Mm-hmm. So those are the three key pieces if you're going to go forth and take advantage of the new technology that exists to link you to the new treatments. Definitely, and especially this overexpression piece. Mm-hmm. That's what we're seeing in terms of finding the best options and making sure they're most targeted to you.
Thank you for watching CTOM TV. Subscribe below and stay informed.